his review on cancer as an evolutionary and ecological process from, when was it? 2006. Must be one of the founding documents of this whole field. And he will tell us more about his research and I hope about his society on evolution and ecology.
Yeah, I thought I should put this up. I'll put this up at the end of the talk also. But there is a society that I encourage you to join. It hosts a conference every other year on evolution and ecology of cancer. Thank you for inviting me. It's a real pleasure to be here. What I thought I would talk about today are what are the most important clinical problems we have and how does an evolutionary and an ecological approach to those problems, or how can we approach those problems from an evolutionary and ecological perspective. So for me, I believe the 5 major problems we have in oncology are risk stratification. So in most cases for a pre-cancer, we want to know, is this something that we can ignore or is it going to be clinically relevant, i.e. likely to kill the patient? That's true for predicting progression from a pre-cancerous condition to a cancerous condition. We'd also like to know if you have a full-blown cancer, how dangerous is it? In some cases like prostate cancer, you may be able to just watch it. Other cases, you have to intervene immediately, and how to intervene are important. So we need to stratify high versus low risk, medium risk. That's a broad problem. Another, of course, then is cancer prevention. So if you have a high risk pre-cancerous condition, you'd like to do something about it to prevent it from becoming cancer, or in general, have a public health impact by preventing cancer. It's one of the big problems we have. Probably all of you know that the major determinant of whether you can cure a cancer or not is when you detect it. So there's a lot of effort in early detection, because if you can detect it before it metastasizes, often you can surgically remove it. And something that students should know is that surgeons cure a lot more cancer than oncologists do, dramatically more. And it's because of this point. Then of course, if you can't cure it, you would like to have some kind of effect on the tumor, so therapeutic response, which we usually think of as just as shrinking the tumor. That's one of the big problems people worry about, particularly in pharma, when you're developing a drug. And then I think the most important clinical problem we have with full-blown cancer is therapeutic resistance, that we apply a drug, we select for resistant clones, or change the phenotype so it becomes resistant, and then we can't save the patient. And that's the short description for why cancer is still a problem, that we select for therapeutic resistance. So I think these are the 5 big problems, and I submit to you that they're all either evolutionary problems or that evolution has a way of addressing these problems. So I'm going to take you through, I was asked to sprinkle in the results from my lab here, but this is sort of more of an overview than just particular research results. So first of all, risk stratification. This is the area I think that we've worked in most among these 5 problems. And one big issue we have in cancer, of course, is that tumors are spatially heterogeneous. So typically we take one biopsy from a tumor, but we may pick up the wrong clone, the wrong bits of the tumor with respect to what's going to happen to the tumor in the future. And so this is a figure from Marco Gerlinger and Charlie Swanton's paper in 2012 in in New England Journal of Medicine. And they did multi-region sequencing, and in this case, this is a panel of expression profile for the different samples from the same tumor, in which they found some very high-risk areas and some very low-risk areas. And just that basic result is a dramatic illustration that if you take a single biopsy, you often get the wrong answer, or you can get the wrong answer, because of the spatial heterogeneity. And it turns out that no matter what assay you use on pretty much every tumor you find intra-tumor heterogeneity, variation, diversity within the tumor. So this is a fundamental phenomenon in cancer, and it's what drives, of course, natural selection. You have to have variation within a population in order to get natural selection, so it's also driving the dynamics of cancer. Another serious problem here with risk stratification is that there's this long tail of rare clones. Another way I think about this is that evolution itself is driven by fitness outliers. And yet in oncology, we typically measure the dominant clone to whatever is the majority in the sample you have, whereas the future of that tumor is likely to be driven by a very rare clone. This is some data. This one on the left is in ALL, so a blood cancer, and I don't know how well you can see this, but they did 10,000-fold sequencing on just the one IG locus, and so you can get frequencies of clones down at the 0.0007, 0.0002. You find, no matter how deep you go, you'll continue to find these rare clones. So that's a big problem for our measurements. The other data here is from the normal skin of the eyelid that Inigo Martin Correa sequenced, and of course picked up lots of tiny clones in the eyelids of these people that were getting eyelid reduction surgery. So there's a very long tail of rare clones. It's a big problem for us. And this is also one way that I think about some of the challenges of doing risk stratification, or in particular biomarkers. I wanted to take you through these. So these chimerograms, we call them, describing how clones change through time, help us think about this process. So in this upper case here, I can't see my biomarker, I can't do that. You can see that you might have a biomarker which we're going to call X, like presence of a mutation like P53, maybe a gene expression profile, whatever. Okay. So if you take a single biopsy, you may do it in the upper left-hand case where you miss X, just because that's spatial heterogeneity, right? In the case B, you take a biopsy, but then the critical event of X occurring happens the next day, the next year, whatever, because this temporal heterogeneity, because things are changing through time, right? So you, again, would get a false negative in both of those cases. In panel C, you get a true positive, so you hit the clone, it has the marker, but because neoplastic progression and carcinogenesis are stochastic processes, the remaining events necessary to generate a cancer in this case never happen, and so you get a false positive actually, in terms of predicting cancer. And of course, one thing we've learned from a lot of the sequences we've done is that there's many different ways of generating a cancer. So you've got your biomarker X, but maybe this cancer is progressing through a sequence of Y and Z and never had X, and so in this case you get a false negative, right? So all different ways in which it's very difficult to predict these evolutionary stochastic processes. So instead of looking at the products of a stochastic process, we argue that we should be looking at measuring the process itself. So here are the parameters that affect the rate of evolution, or the evolvability of a tumor, a population. And our hypothesis is that by measuring the evolvability of a tumor, we should do better at risk stratification. So mutation rate, population size, generation time of the population. Notice the population here is not every cell in the tumor, it's every stem cell in the tumor. There's some controversy about what we mean by stem cell, but I just mean cells that have indefinite replication and can propagate the population through time. And we don't have good measurements of the generation time of cancer stem cells, so this is an issue. And then rate of clonal expansion, rate of clonal expansion is how a cancer biologist would say it. An evolutionary biologist would talk about a selective coefficient, or maybe selection intensity that Jeff was talking about. Yeah, Eva. So Eva's question is, why am I specifying stem cells and not just all the cells or normal cells? Because selection acts on all the cells. So the theory is that some cells in the tumor have a limited proliferative potential, so maybe they can only divide 5 times. Right, so those are evolutionary dead ends. But some cells in the tumor are indefinitely self-renewing. Right? And so if you want to ask what's going to happen to the population of the cells in the tumor for a year from now or 2 years from now, if it's true that some of the cells have limited potential to divide, they're unimportant. Now that's controversial because there's some evidence that, quote, non-stem cells can change state back into a stem cell. So in that case, those cells are actually relevant to the future of the tumor. And it's different probably for different types of cancer. So in melanoma, it looks like every cell is essentially a stem cell. But in some of these leukemias, it's 1 in 10,000. It's a mess actually because we use a operational definition that has to do with engraftment in a mouse. And so if you use a different mouse, you get different answers, things like that. So I don't want to get into too much of the mess, but as an evolutionary biologist, what we care about are the population that is going to be relevant to the future of that population, the individuals that are relevant to the future of that population. Yeah, Eva, you don't look, yeah. All right, so sorry, so your question is it possible for the daughters of these non-stem cells to become like stem cells, right? Well, okay, so I think you're using a organismal depth understanding here. So when a cell divides, it doesn't remain, right? It makes two daughter cells. So if a cell is not a stem cell, what we're saying is that both of those daughter cells and all their daughters have a limited proliferative potential and will stop. Right? Now, if it's the case that they can become like stem cells, then yes, they're relevant.
Carlo, can I just add something that sort of adds to this discussion and takes you a little bit away from the stem cell debate, which gets very contentious, which is just that when in cancer, obviously there's a lot of, it depends on the cancer and the type, but there's like necropsy, there's a lot of death of cancer cells as well as the survival of cancer cells. And in addition to a lot of death, there's probably a lot of cells that are moribund or not producing very many daughter cells and not likely to produce daughter cells that will produce daughter cells just because they have mutations and chromosomal abnormalities that are not viable. So even if you completely discard the stem cell concept, there's still a lot of cells in a tumor that really don't have a future. They're dead ends. And so from that standpoint, we don't know the population size unless we know that quantity.
Right, so this is all open questions. And so the hypothesis would be if you have a measurement of stemness or of propagation potential, that the number of cells that are high on that potential should be a predictor. It might not turn out that to be true, but there is some evidence that measuring the number of stem cells is relevant clinically. All right. We've done most of our work measuring diversity, that intra-tumor heterogeneity, and I'll show you some results. We should also be able to get relevant information by measuring what are the selective pressures on a tumor, because of course that helps. If you understand the pressures, you can make pretty strong predictions about what's going to happen to the phenotypes in the population. All right. So here's some recent results in Barrett's esophagus, where we were able to take single crypts, so single clusters of cells from this pre-malignant tissue, and measure them genomically, in this case with SNP arrays. And we get multiple time points from the same patient, so you can look through time and you can start actually estimating the mutation rate. Mutation here, copy number changes and loss of heterozygosity changes. And what was an interesting result of this was that we could see an increase in the mutation rate by orders of magnitude prior to genome doubling, and then a continuation of increases in mutation rate after genome doubling. So that was, we didn't know that in the literature before this, but there was a hypothesis that you needed to have genome doubling to get this increase in mutation rate. It does not seem to be the case in Barrett's esophagus. All right. Here's some early work that we did in Barrett's esophagus. So at baseline, you get a measurement of multiple biopsies from the same tumor. You can calculate the amount of diversity, and what we found is the upper quartile, people with more diversity in their tumors are much more likely to get the cancer, esophageal adenocarcinoma, than the people with low diversity. And that was just with a small microsatellite panel because it was measured back in the late 90s. And some of those microsatellites show you lesions in P53 or P16, these tumor suppressor genes. So you could ask, well, what if you only define diversity based on driver genes? That's predictive. But if you define diversity entirely on neutral loci, that's also predictive. So measurements of diversity or intra-tumor heterogeneity are actually relatively robust to how you do it. And exactly what statistic you use for that diversity, whether it's the Shannon index, which is just entropy, or the Simpson index, which is the likelihood that two samples come from the same clone, or just how many different clones do you find, which ecologists call species richness, those were all predictive of progression to cancer. So one reason we're excited and think this is a good path for the field is because of this robustness. That it doesn't matter what you use to measure diversity, it doesn't matter what statistic you use for diversity, give or take a little bit. They all seem to work well, at least in Barrett's esophagus and in a number of other diseases. Another thing that you should understand in cancer biology in general is that we don't change clinical practice unless you can show something works independently in different cohorts. So our initial results were back in 2006, and I was excited that 10 years later we finally were able to get confirmation of these results in an entirely different cohort with entirely different assays, but again, diversity in Barrett's esophagus predicted progression to cancer. Okay. I think I'm going to skip this in the interest of time. I wanted to show you this result, and I talked a little bit to Jeff about this the other day, because it's an open question why we see this. So in this study, it was a pan-cancer study, and we asked if you estimate the clone size within your sample, you can say, well, mutations in CDKN2A up there, in bladder cancer, when they showed up, they tended to show up in 91% of the tumor sample. So very large clones, mostly drivers. I mean, mostly trunk clonal mutations. And you can rank the different cancer genes by how large of a clone do they generate when they happen. So given that they've happened, how much of the sample contains that lesion. So one of the things we just discovered and didn't expect to see ahead of time is in that Kaplan-Meier curve that if a tumor has a mutation that typically shows up only in a smaller clone, those were more deadly than tumors that only had mutations that showed up in larger clones. Right? It's an open question why that is. So my hypothesis is that the mutations that tend to generate large clones are mutations that have a big selective effect, and so it's relatively easy for the population to discover those mutations and for them to expand. Whereas if you have a tumor that has not only discovered the easy things, but the less easy things, the harder things to discover, the mutations that have smaller effects, that tumor has explored a lot of genomic space and is probably very diverse and very evolvable. That's my guess, why we have this result. But it's an open result. Okay, what we did in this pan-cancer study, among other things, was using this expands package from Noemi Andor to estimate the number of clones within the sample, and to show that tumors with more clones had a worse prognosis than tumors with fewer clones. So this basic diversity predicts overall survival. I want to point out to you that in general we don't know the dynamics of the clonal dynamics in cancer, so we can get these snapshots, typically only one time point per tumor, off typically only one biopsy per tumor, which I think is awful for doing this evolutionary work. But we don't know how fast clones expand or contract, how often new clones show up. So there's a lot of open questions about the fundamental dynamics of cancer from this perspective. And we don't know how that diversity is maintained. One long-term question in evolutionary biology is how is diversity in a population maintained, and the same thing is going to be relevant for cancer. So if you have two clones that actually are cooperating, then you can have stability of those clones. That's one possibility. You may have parasitism type interactions or niche specialization, because there's different microenvironments in tumors, and so those should produce different selective pressures, and you might get clones that are specialized in some of those niches. These are all open questions, hopefully for you guys. Okay, so that's the risk stratification side. So cancer prevention, if you understand this process of neoplastic progression and carcinogenesis, we would like to intervene in that process, right? So that's where we come in here. And again, given that we could maybe measure the dynamics of evolution, we can then start targeting those dynamics. So can we develop a cancer prevention approach which is designed to reduce the mutation rate, reduce the number of stem cells, lengthen the generation time of those stem cells, and slow this whole process down. And an important point to get here is that the whole process from the first event in carcinogenesis, which is called initiation, up to the last event called transformation, when a cell becomes fully blown full cancer, that has been estimated to take, in some cases, 5 to 6 decades, so 50 to 60 years. You can see that in breast cancer and colorectal cancer. Sometimes it's short. You can see sometimes people get an extreme exposure, radiation burst, carcinogen, and you can see it takes only a couple years. So estimates of the process of neoplastic progression range from 3 to about 60 years. But that also implies that if you could slow down this process by just a factor of 2, not even an order of magnitude, just a factor of 2, then in many cases the process of carcinogenesis would take 120 years. Right? So I think a important and viable research program is just slowing down the process, not preventing cancer by killing all the cells or stopping it dead in its tracks, but just reduce the dynamics by a little bit, and then most people would not be dying of cancer. Similarly, you could ask about just changing selective pressures and sort of select for the kinds of cells that won't kill us. This graph here is from one of our papers where we showed that in Barrett's esophagus, we would take 2 time points when people were not taking NSAIDs. The NSAIDs are non-steroidal anti-inflammatory drugs. You know them as things like aspirin. So 2 time points when they're off aspirin, 2 time points when they're on aspirin, because usually for their heart, and found that the mutation rate dropped by an order of magnitude when they went on aspirin. And that's what, these are 13 patients, and those estimates are the mutation rate up there. Okay. I think I basically already said this, that there's different ways of slowing evolution by targeting the different parameters of evolution. Okay. I wanted to show you some fun stuff that we haven't really addressed yet, which is in actually comparative oncology. And the point here is that evolution, in some cases, has actually solved this problem of cancer prevention before. And this brings us to Peto's paradox. So Sir Richard Peto is an epidemiologist who is famous for developing evidence that smoking is associated with causing cancer. And he pointed out in the 70s that a mouse has 1,000 times fewer cells than a human and only about a 2-year lifespan. And so if all things were equal, if a mouse had the human biology, a mouse would never get cancer, virtually never, right? The interesting actually side of this is projecting in the other direction. An elephant has 100 times more cells than a human, roughly the same lifespan as us. So if they had human biology, they'd be getting a ton of cancer. Maybe they'd be going extinct because they get cancer before reaching age of sexual maturity. But of course they don't, because there's been selection on elephants to suppress cancer long enough to grow large. Larger elephants, you should know, have more mating opportunities and higher social status. And so there's a lot of selection on elephants to prevent cancer so they can get large and make more babies. And this brings us to this graph. So one of the things that we're having a lot of fun now is we've collected up data from a variety of sources. We have over 170,000 pathology records of animals, and so we can estimate what's the cancer rate across species. And so this is an initial graph of a number of species. We have, I think, over 2,000, where we have enough data to start estimating cancer rates, or neoplasia rates in this case. The overall statistics you can see in the upper corner. Mammals have a lot more cancer than birds and amphibians and reptiles. And so we're very interested in what is the biology behind organisms that have very low cancer rates, and also organisms that have very high cancer rates, like mouse lemurs. And we're analyzing this in terms of life history theory. So life history theory is about the ecological conditions that select for slow reproduction, investment in maintaining a body, et cetera, like an elephant or a human, versus fast reproduction with relatively little investment in somatic maintenance, like a mouse. So here's a few little highlights of this. So we've got over 1,500 dolphins in our database, and they get very little cancer, about 1.8%. The poison dart frog also is just very little cancer, not so many. We have 175 cases of that in our data, and only one of which shows any kind of neoplasia. Hummingbirds also, 62 hummingbirds in our data set, 0 neoplasia so far. So if you just pull out the mammals here, one thing you can see here is actually the opposite trend than you would predict just from a pure cancer biology perspective. So having more cells and a longer lifespan is actually associated with having less cancer. Right. And it's because of that selection, to reduce cancer in large animals. I should also say that one important aspect here is that this is zoo data. These are animals in captivity, right? And most of the animals on this side of the graph, these fast life history animals, would get killed by a predator in the wild. So if you protect them from predators by putting them in a zoo, what you end up doing is revealing that they have very poor cancer prevention biology. Whereas animals on this side of the graph, they're already too large in general to be having a lot of external mortality like predation. And so when you put them in a zoo, you don't change that very much, and you reveal the fact that they actually have quite good cancer prevention. So you can see the same trend in birds. Chickens have a lot of cancer, by the way, that's ovarian cancer, so we think that has a lot to do with selection for very high egg production. Reptiles, also relatively low, and alligators are the outlier here, so we don't know why alligators have such good cancer prevention. But we did a study in elephants and discovered, initially genomically and then verified in a wet lab, that they have amplified p53. So instead of having 2 alleles of p53, they have 40 alleles of p53. We just cloned it out. And along with that, they have extremely sensitive DNA damage response. They're no better than humans at repairing DNA, but they're much better at humans at killing off the cells that have some DNA damage. So these are just those results, humans versus elephants, with various grades of radiation. And we also know that this is happening through a p53 pathway, but it is not the case that these extra copies of p53 are just acting as redundant alleles. In fact, most of these copies are missing critical bits of p53. And our colleague, Josh Schiffman at Utah, has shown that the biology of what's happening is actually quite different. The p53 is not acting within the nucleus, but actually getting out into the cytoplasm and then into the mitochondria and triggering apoptosis inside the mitochondria. That's ongoing work, hasn't been published yet. Okay, this of course, if you're talking about big animals, you have to start talking about whales, so we're sequencing the humpback whale at the moment. And doing the comparative analysis. This is my postdoc Mark Tallis, who's just got a position at Northern Arizona University. And I just want to give you some highlights of this because it's not yet published. But you can do various comparisons within this phylogeny of cetaceans and find things about evolution, signals of evolution and Wnt signaling and DNA repair and double-strand breaks, etc. So then you can put that into a Go analysis and identify various pathways or phenotypes that are evolving, having to do with cell proliferation and double-strand break repair. So that actually is similar to the results we're getting with elephants, just through a slightly different mechanisms. We've teamed up with Thomas Marquette Bonnet and his postdoc Lucas Kuderna to analyze the humpback whale DNA for copy number, segmental duplications and copy number changes. And actually did find a spike here. Those little tick marks at the top are where the exons are for these different genes. And you can see the copy number graphs below, and you can see that about the first 3 or 4 exons of p53 are showing a high copy number even in the humpback whale. So actually, maybe some similar mechanisms in whale as well as elephant. So I'm excited about this because large body size has evolved independently multiple times in the history of life. And so it's quite likely that you're getting different solutions to the problem of cancer prevention, and the answers are just out there for us to discover. So that's fun. All right, what about early detection? I'm running out of time. So you wouldn't think that evolution had to have much to do with early detection, but actually when we started asking about what's the fundamental nature of cancer, cancer is a disease of multicellularity. So so there was no cancer before the evolution of multicellularity, so about 1 to 2 billion years ago, there was no cancer. But then by 500 million years ago, there's all these different organisms, and so cancer becomes a problem. But that means that cancer should be a problem for every form of multicellularity. Now, multicellularity has evolved independently 7 times in the history of life. So once in animals, twice in plants, twice in fungi, once in red algae, and once in brown algae. So we looked at what does cancer look like across all these different branches of multicellularity. But it forces you to ask, what is cancer? Because cancer is defined as an abnormal growth that invades through a basement membrane or goes to a different part of the body, metastasis. But most of these organisms don't have a basement membrane. So it's a totally anthropocentric definition of cancer. Right? So it actually forces us to ask and redefine what do we mean by cancer. And we do this through cooperation theory, and we argue that in order to build a multicellular body, the cells have to cooperate on these 5 parameters. They have to stop proliferating, the somatic cells have to stop proliferating. They have to coordinate cell death. They have to allocate resources through the body, maintain the extracellular environment, and do some kind of specialization or differentiation, the division of labor. And then you can understand cancer then as cheating on these forms of cooperation. And that's a general definition that can apply to all these organisms. But it also then implies that you could start doing early detection by looking for cheating on these forms of cooperation. And it leads you to new proposals, which is you could have a pre-cancer that is not a tumor, that is not a mass, because it has not yet started cheating on these uncontrolled proliferation or apoptosis suppression, but it has started cheating in terms of resource monopolization, loss of differentiation, and other things. So it leads us to some novel predictions about detecting either pre-malignant or tissue that's starting to become malignant. So that's all I'm going to say about that. Therapeutic response, we did a study asking what causes a species to go extinct, because it's analogous to driving a tumor extinct, right? A species is a genetically diverse population spread over some geographical area, and the tumor is a genetically diverse population spread across, in the worst case, geographical areas of the body. And so we'd like to understand how we could drive a tumor extinct. What you learn by studying species extinction is that you need more than one selective pressure. Usually there's what we call in paleontology a press-pulse dynamic, where a population is stressed, it's pressed by some selective pressures, and then there's an acute change that drives them extinct. And those pressures have to be very independent and maintained for, in the case of species, thousands of generations. Now, when we do chemotherapy, we do not apply a therapy for thousands of cell generations. We often apply it for one cell generation. So what we're doing in therapy is quite different than what drives a species extinct, and may explain in part why we've had so much trouble doing generating cures. I'm going to skip this also. If you're interested, you can read the Nature Reviews Clinical Oncology paper. The most important bit I wanted to cover and end with is therapeutic resistance. So it's a relatively simple process to understand. In general, there's diversity in the population. Often there's so much diversity that when we apply a drug, there are some mutants in the population that are resistant to the drug. So we kill everything else, the tumor grows back from those resistant cells, and now the entire tumor is resistant and we can't use the drug anymore. That applies to every drug that's ever been invented for cancer. So what we should be doing is thinking about what do we want to select for. We're selecting for resistance. It's obviously not what we want to select for. So we should be thinking evolutionarily, how do we manage this evolving population? And there's a bunch of possibilities, but an important aspect of this is that we've been blinded into trying to kill cancer cells. And the ultimate objective of therapy is not to cure cancer. The ultimate objective of therapy is to preserve life. And the fact that we've been focused on killing the cancer cells has meant that we haven't been paying attention to ways of living with cancer or preserving life, right, in other ways. So Bob Gatenby had this idea from pest management, where they have the same problem. You spray a field with pesticides and you select for resistant pests. They've learned that the best way to manage resistance is to keep sensitive pests around, to not go for eradication, not go for a cure, but just try to maintain control over the population. And the way I think about this is that oncologists are able to control sensitive cells, and sensitive cells, it turns out, are able to control resistant cells because resistance has a fitness cost in most cases. It's an open question how much or how often, but in general, there's some cost to being resistant. And so in the absence of drug, sensitive cells can outcompete resistant cells. What we typically do in therapy is we kill all the sensitive cells, there's nothing left to control the resistant cells, and the resistant cells kill the patient. So Bob Gatenby's idea was, what if we just try to keep the tumor at a stable size, try to maintain sensitive cells there, and thereby be able to indirectly keep keep control over the resistant cells? So in mouse models, this works really well. It's called adaptive therapy, and you can keep the tumors under control indefinitely. There's 3 different types of cancer, a number of different experiments, and they had to sacrifice the mice for old age eventually in that arm. Whereas the other conditions, high-dose therapy, you lose control and it kills the mice. There's now been the first clinical trial in castration-resistant metastatic prostate cancer. The idea here is that you withdraw the drug, you go on a drug holiday before you've killed all the sensitive cells. So in the case of prostate cancer, if the PSA level drops below 50%, then you stop dosing. And that's the black bars on the upper graph there. Whereas the standard of care is just to keep these patients on the drug, abiraterone, continuously until the tumor starts growing again. And that's the lower bars here, which is the control condition. So historically, in this condition, it takes 16 months before half of the people have started, their tumors have started to grow again before progression. So the median progression time is 16 months for this condition. This was published when only 1 of the 11 patients had progressed, and they are out to, I believe it was 24 months. Now we're out to, I think, 37 months. Still only 1 patient has progressed, and none of the other ones have progressed now. And if everybody progressed now, then we'd have a median progression time of 37 months. You can see the p-value was already tiny at that point. So this is a pilot study, so what has to happen now is a larger study to confirm it. I'm going to skip this also. So the reason I'm so excited about adaptive therapy is it's the first time that we have an evolutionary algorithm, first time we have any algorithm for managing therapeutic resistance. In oncology, we don't manage resistance, we just give up on a drug and try another one. Right? But this is a theoretically based and now experimentally justified approach to actually controlling the tumor. And it's a way of changing an acute lethal disease into a chronic disease that we could live with. Right? So I didn't go into this because everybody else has, essentially, that evolution is the theory of cancer, somatic evolution. But we have a lot of tools from both the evolutionary ecology for studying and also intervening on cancer. So there's huge opportunities, and it really helps to solve the big clinical problems in cancer. So I just wanted to thank my funding and collaborators, and I said I'd put this back up here, but I invite you, please join the society and come to our conferences. We'd love to have you. Thank you.
Yeah, okay, so we were actually discussing this yesterday, this adaptive therapy, and I'm still kind of, so the problem that I have with it is this, that many studies suggested, actually also in breast cancer, that there is this early emission of metastasis. So even small tumors, if you would manage to keep everything small, and even if you do this in metastatic cancers, you already have metastasis, you keep them also small, but then they will, according to what we heard also 2 days ago, they will start spreading around. So then at least you need to keep adjusting these dosages that will keep this burden somehow controlled. But I don't imagine that this will work forever, right? These metastases will spread and spread and spread and spread, right?
Well, okay, maybe is the quick answer to that. The measurement in prostate cancer was PSA level in the blood. So it captured, we're measuring the total tumor burden regardless of whether you're getting more and more metastases or whether you're getting fewer and fewer, right? So we're not missing that dynamic. So the evidence so far is that it is working. Now, another important point to know about cancer, particularly for the students, is that the doubling time of a tumor is often measured on the order of months, whereas the doubling time of cells, particularly in culture, is measured in usually a day or two. And in general, our interpretation of this is that there's a lot of cell turnover, that the tumors are not expanding without constraint. Right? There's probably over 90% of the cells are dying in general, and so the total population is growing slowly. My worry about metastasis is that it leads to a loss of constraints on population sizes. And you need that competition between sensitive and resistant cells to keep the resistant cells under control. If all those cells are growing without constraint, then there's no competition and you can't keep a lid on anything. So I take your point as, once you have metastasis, it's not clear this would work, right? But the evidence is that we did this in a metastatic cancer and it is working, and it's probably because there are lots of constraints that are forcing a lot of competition. Aika? Right. So let me just, okay, go ahead. I'll repeat it after you finish. Sorry, which diagrams are you talking about?
It's working, the signal path.
walls and transmitter is inconveniently located
no.
there in the corner, so just try again and have them.
You mean the diversity versus survival?
Like this. Okay. Try that, but don't touch, don't touch.
Like that? Right. Yeah, no, that's fair. So a lot of these diagrams imply a constant population size, which actually is true in Barrett's esophagus. It's a constant population, but in most tumors, yeah, it's growing. And so I think the better, you've seen previous talks with these chimerigrams where actually you can see the whole population growing over time as well as the proportions changing. So yeah, fair. Hey, Russell.
So I was kind of intrigued. So I was intrigued because this model of adaptive therapy you're describing seems very similar to some of the ideas of what's going on in immune surveillance. So why tumors are, in many cases, the immune system is microscopic tumors rather than wiping them out. And I think we'd usually assume it's just that the immune system isn't efficient enough or effective enough. But now I'm wondering, maybe the immune system has evolved to do this naturally.
Yeah, so we think that immune system in general for pathogen challenges doesn't get activated until it rises above some level of danger, right? And the same thing as we've learned to do in pest management. We don't actually treat fields until the pests have risen above some level of danger. And then we'd knock them down, but you don't actually have to eradicate them for the patient to survive or for the field to produce good crops. So yeah, it may be the case that for the similar reasons, there's some diminishing returns, and it's probably better to tolerate some level of problem, right, rather than spend all the resources necessary to completely eradicate. Or yeah, so maybe. Yeah. Right. Right. So, Valentina.
Thank you.
So the approach you presented will work on some patients, and some patients it will fail. What would be your guess now, before you did all these clinical trials, what parameters in the patients could predict, in theory, for which it will work and for whom it will fail?
Yeah, there's actually been a fair amount of mathematical modeling of this. So if the proportion of resistant cells is already large, this typically doesn't work. Of course, the first parameter is, can you get any response at all, right? So sometimes when we treat a tumor, we don't kill anything, right? Because this drug doesn't work. So this strategy can't work in that case. So these clinical trials are set up such that one of the entry criteria is that on the initial doses, the tumor shrinks. That's the first thing. But if it only shrinks by a little bit because 90% of the cells are resistant already, you probably can't play this game. There were studies in prostate cancer actually on intermittent therapy, which is similar. You stop dosing after some time and dose later. It's not the same for one crucial reason, which is that the intermittent therapy wasn't conditional on what was happening in the tumor. So this algorithm is conditional. But those trials were not successful. The intermittent therapy, they had no improvement. They weren't any worse than the constant therapy, so there's some benefit in terms of less drug, but they weren't any better. But it turns out that they applied the drug for 8 months at high dose before they started doing the intermittent therapy, and our models suggest that that was too long. They killed all the sensitive cells during that first 8 months, and so they couldn't play this game.
Yes.
Yeah.
Sorry, in the adaptive design that you mentioned, the direction of the time that you quit up the drug is patient dependent?
Yes.
So you have a mixed random effect model or something from longitudinal data?
So yeah, so the algorithm in the clinical trial was if PSA drops below 50%, or when PSA drops below 50% of its initial level, you stop dosing. And when it comes back past 100%, you start dosing again. So one thing I like about this is that it's personalized medicine, because some patients, that cycle going below 50 and coming back took 2 years. In other patients, it took 2 months. So you saw very different dynamics. And yeah, you have to treat the patient. You treat the patients individually, and rather than having one script that you hope would fit everybody.
Don't want to cut the discussion short, but we're running a bit late, so let's thank Carlo again.